All right, let's take a look at ribosomes and protein synthesis. So yesterday we looked at the formation of messenger RNA in the nucleus and how messenger RNA copies the code from the DNA, and that will ultimately be the code that will be translated into a protein. So today we're going to look at how that code works, describe the role of ribosome in assembling protein, and then, some terminology of how molecular biology relates to genetics. The genetic code. So what we're looking at here is a section of RNA. We know it's RNA because it has uracil in it and it's single strand. All right. So when we're looking at what we're ultimately coding for is a protein, and proteins are made up of amino acids. The code for the amino acid is held in these three A's, U, G, for example, right here, which is the start codon for the beginning of translation. So those three nucleotide bases is called a codon. So for one amino acid, you need three possible nucleotides, A, U, G, C. How does that work? To read it, we're looking at that's A, U, G, A, C, C, obviously U, C, U. So that's the code. Now, that will code for different amino acids. And because there's three, we're going to look at 64 possible three-base codings. Now, why you start is the first number, G, C, U. So what we had was, back here, AUG. So A, U, G is the code for methylene. C, U. Do we have another one back here? We didn't have a C anything. All right, so they all code for different amino acids. UUG, UUA code for leucine. UAA is a stop codon. UAG is a stop codon. So we have ones that are start, that AUG is a start codon. And then the ribosome needs to know where to stop reading the code. So we have those. And UG, G is tryptophan. So there's different amino acids are coded for differently. So as it says, start in the middle, work outward. And there's your histidine, a CAC. Methylene, AUG is our start. I already said that. And then again, UAA is stop and UAG is also stop. Okay. So we have transcribed the message from DNA to RNA, and now we're going to translate the message from RNA to a protein. Okay, so transcribed mRNA directs the translation problems. Translation is the process that produces proteins by decoding the sequence of an mRNA codon. So there's our mRNA. We look at the nucleus, and the nucleus has these pores in it. So what we're talking about is how this works in a eukaryotic cell. In a eukaryotic cell, the mRNA is produced in the nucleus. Okay, let's talk about translation. So how do we get the code from the code to an actual protein? Well, the ribosome is going to attach to the mRNA molecule. And so this is the mRNA molecule. And the mRNA molecule will come along and will read, and it gets to this start codon, AUG. Okay. Then we have another one called the tRNA molecule carrying amino acids that bind them. They bind to the mRNA codons. So this tRNA has a corresponding or a matching code. So if the mRNA is AUG, the transfer RNA is going to have a code of UAC. So they bind like that. The transfer RNA then carries this methylene, which is the start molecule. And then we have phenylalanine. UUC codes up at AAG, and that's phenylalanine. So now the ribosome is going to take these amino acids and they will be put together in what's called a peptide bond. Okay. This lysine is coming in. AAA will bind with UUU. Codons, anticodons. So this is what we call translation. Translation is the polypeptide assembly. So the ribosome helps form a peptide bond, peptide being a protein bond. It breaks the bond, holding the first tRNA molecule to its amino acid. So here we go. Boom, there's the bond. And this goes off and bonds with another amino acid. And so what we do here is we start building a protein. And its translation direction is from the AUG in this direction. So once the ribosome reaches a stop codon, releases, Yeah, here we go. So once the ribosome reaches a stop codon, it releases that newly synthesized polypeptide. So this is our polypeptide. Poly, many, peptide is the bond between the two amino acids. So many bonds, polypeptide. That mRNA molecules then is completing the process of translation. So what we have is a string of amino acids. Okay, so it hits AUG, puts it puts that on there. And so all these forms of RNA, messenger RNA, transfer RNA, and rRNA are involved in this process of translation. Messenger RNA, again, carries the instructions for polypeptide synthesis, many peptide bonds, from the nucleus to ribosome in the cytoplasm. This player is the transfer RNA. It carries the amino acids to the ribosome and matches them to the coded mRNA message. And then the ribosomal RNA forms an important part of both subunits of the ribosome. All right, so molecular genetics. We're going to see this graphic here. So what we're looking at is the nucleus right here and the DNA right here. And the DNA is translated to mRNA. The mRNA is translated, transcribed to the mRNA. The mRNA is translated to a protein that's made from peptides, from amino acids that make a peptide bond, make a polypeptide chain. Okay, so this is what we call the central dogma of molecular biology, is that information is transferred from DNA to RNA to protein. When I say central dogma, I mean this is the thing, the central dogma, okay? This is the way it works. When a gene of DNA code is used to build a protein, we say that that gene has been expressed. So gene expression. Proteins are made, we have a lot of proteins in our body. They're big, complex. Those peptide chains then have multiple charges on them and shapes, and they form a multi-shaped, three-dimensional, they form a three-dimensional structure that forms a specific function, such as lactase. So we express a gene called lactase, which is a protein that breaks apart lactose when you eat lactose. So you probably all know someone who's lactose intolerant. Well, we produce this enzyme when we're young. We express this enzyme when we're a baby so we can digest milk. But at a certain point, evolutionarily, we're not walking around finding milk most of the time. It wasn't until domestication. Some people produce it. Some people just turn the gene off. Some people turn the gene off and turn the gene back on again. That's called gene expression and epigenetics. So there's things that trigger the gene to turn off and the gene to turn on. However, gene expression is just meaning that that code is being turned into a protein, okay? So again, DNA to RNA. The RNA is translated then into a protein. So that is that in a nutshell.